are you? Today we are going to start with a new unit dealing with the immune system. In this presentation we are going to cover the first activity in chapter 6. It's going to be on your official book, page 112. First of all, we're supposed to know what is the immune system. So it is one of the systems of our body, but this system is a very important system that deals with the protection of our body from any foreign substance, from any foreign cell or tissue. Of course, when it detects any non-self element, so any element that doesn't belong to the body, an immune reaction or an immune response is going to be carried out. This immune system is characterized by the ability to distinguish between the body's own components, which are said to be the self, and the foreign elements which do not belong to our body, and they are said to be non-self. We're supposed to know what is meant by the HLA, or a major self marker. In order to know the HLA, we're supposed to carry out certain experiments, basically skin grafting experiments. So if you take a look into this document, we've got here a mouse, and in which a skin was grafted into another mouse having the same strain, and as you can see, there was no rejection after 10 days, which means that this graft was accepted. On the other hand, we have two different strains of mice, in a way that one of the two mice was the donor of the skin, which was grafted into the other mouse of a different strain, and as you can see, after 10 days this graft was rejected. What are the graft types? First, we have the autograft. Autograft is grafting or transplanting an organ or a tissue from one part of the body to another part, but in the same body. Usually this graft is accepted. On the other hand, we have the allograft, in which it is the transplantation of an organ or tissue from one organism, from one body, to another organism, but they both belong to the same species, and this graft is usually rejected. We also have the isograft, which is the transplantation of an organ or a tissue from one organism to his or her identical twin. Of course, this graft is gonna be accepted. We also have another type of grafts known as the xenograft. It is a graft done between two different species. Of course, that type of graft is usually rejected. But what makes the graft accepted and what allows it to be rejected by the body? To know or to answer this question, we're supposed to know that on the surface of our cells we have specific markers which are known as antigens. These antigens are glycoproteins in nature. That is to mean they are made up of a mixture of proteins with a carbohydrate or a polysaccharide. These antigens, they form actually the biological markers. They represent the biological identity card for each cell. So now, because each cell has this biological identity card, this makes it clear for our immune cells to be able to distinguish between those self markers and the non-self markers which do not belong to our bodies. These antigens are referred to as the HLA molecules, or the human leukocytic antigens. Usually these antigens or these HLA molecules are coded by specific genes known as the MHC genes, which are known as major histocompatibility complex. These genes usually code for these antigens, and this MHC system, set of genes, consists of 6 genes, and they are located on the short arm of chromosome number 6. The names of these genes are A, B, C, DP, DQ, and DR. The MHC genes code for 2 HLA classes. The first class is the HLA class 1. It is coded by the loci A, B, and C. We said previously that the locus represents the location of the gene on the chromosome. So the loci is the plural of the locus. So the genes, the MHC genes A, B, and C, they code for the HLA class 1. Usually HLA class 1 antigens are expressed by all nucleated cells of the body. On the other hand, the HLA class 2 antigens are coded by the other 3 MHC genes known as the DP, DQ, and DR, and these HLA class 2 molecules are expressed by some cells of the immune system. That is to mean that the cells of the immune system that express the HLA class 2 markers are going to also express the HLA class 1 markers. So this is a document to show the difference between a marker of class HLA class 1 or MHC class 1, and this is another marker of a class 2, so MHC or HLA of class 2. So this type of markers is present on every nucleated cell of the body, while this class 2 is only present on some immune cells of the body. So returning back to what makes a graft accepted or rejected, we are supposed to know that when a graft is accepted, it is usually accepted due to the convenience or due to the matching between the HLA markers of the donor with those of the recipient, which leads us to know that when these HLA between the donor and the recipient mismatch or they are not matching, this is gonna lead to the rejection of the graft. This is a very important document to show the percentage of the kidney life years. This is a transplant. In this case, it is an organ which is commonly transplanted among humans. It is the kidney, and it is showing the survival rate of this kidney when the HLA between the donor and the recipient match. So as you can see, when the 6 genes match between the donor and the recipient, this is gonna give us the highest survival rate for the kidney, and this rate is gonna start decreasing when the HLA between the donor and the recipient become less matching, or we can say are incompatible. So the survival of a graft and its long lasting in the recipient depends on the compatibility between the HLA markers of the donor and that of the recipient. The genes that code for the HLA molecules, as we said before, are highly polymorphic. That is to mean that each one of these 6 genes of the MHC exists in a very large number of allelic forms. So if you take a look, for example, for the gene that codes for the alpha chain of the DP gene, there are, only for this chain, there are 28 alleles, while there are 138 alleles for the gene coding for the beta chain of this gene. And as you can see, for the other genes as well, DQ, DR, we notice the presence of a big number of alleles, which makes this gene polymorphic and which makes it practically impossible for 2 individuals to share the same HLA molecules, and this is what makes each one of us a unique individual, and this is what actually is the origin of the polymorphism, one of the origins that leads to the polymorphism in the population of the same species, of course. So the HLA, or the human leukocytic antigen, makes it possible for the immune cells to know and differentiate between self cells and the non-self cells. So usually when we talk about self cells, we're talking about every single cell that belongs to our body. So any cell, a skin cell, a stomach cell, a liver cell, any cell that belongs to our body is said to be a self cell to our body, and it is said to be a non-self for any other body, any other human. So usually the HLA forms what is known as a barcode on the, or biological identity card, on the surface of the cell, which makes it possible for the immune cell to differentiate between a self cell, and in this case it's not going to be fought because the self cell is belonging to our body, and the non-self cell, which in this case is going to be fought by the immune cells because in this case it won't be considered as one of the body components. So with respect to this document, we can notice an uninfected cell. Because this is a body cell which is not infected, it is considered of course to be a self cell, and it's not going to be fought by the immune cell. In this case we have one of the immune cells detecting for the non-self. And so the uninfected cells, which appear to be happy, are actually not going to be fought by the immune cell because they belong to the body. They are components of our self system. However, when our body cell becomes infected by a virus, for example, this infected cell will be now recognized as a non-self by the immune cell, and it is gonna be fought in this case. So this is a very important property for the immune cell, which is to differentiate between the self and non-self. Let's take a look into some of the self cells and some of the non-self cells that the body can distinguish. As we said before, any cell that belongs to our body is said to be a self cell. So here we have muscle cell, epithelial cell, a nerve cell, or a leukocyte, which are all said to be markers, or which all carry the HLA markers which belong to our self system. And on the other hand, we have a bacteria, for example a SARS virus, a non-self nerve cell, a non-self leukocytic cell coming from another body. All of these are said to be non-self. Once they enter to our body, our immune system will recognize them as being part of the non-self that have to be fought, have to be eliminated, and in this case the body will be able to distinguish between self and non-self cells. Thank you.